taking a couple minutes to load, but all right.
Mm-hmm.
Is the screen okay this time? Great.
Yes.
Yeah, it looks good.
Yeah, you're in good shape right now.
Fantastic.
Okay, Mark, go ahead.
Okay. Well, welcome everyone to the second afternoon of our Ecology Center lecture series with Dr. Jessica Roy. If you didn't get a chance to attend yesterday, Jessica gave us a really interesting seminar about diversity, inclusion, and engagement with the general public. And those that seminar will be posted shortly, probably next week. You'll be able to find it. So you can check it out on the Ecology Center website if you're interested in that topic. But today,
Mark, let me interrupt you real fast. People can watch it actually on our YouTube channel. It is there from yesterday. I haven't trimmed the front or the end off of it, but if people want to watch it tonight, before I get a chance to trim the front and the end off, it is on the YouTube channel to watch.
okay, so we are a multi-platform center, and you can choose yours. Anyway, today Dr. Roy is going to talk with us about some of her ecology work, the research that she's been doing. I should tell you that Jessica is an assistant professor of biology at Binghamton University in upstate New York. She has a bachelor's degree from Southwestern University, which I'm told is the oldest university in Texas, and a doctorate from the University of Pittsburgh, and where she started working with amphibians, and that led her to some interesting work where now she's works at the border of toxicology and ecology and evolution, thinking about contaminants in a different way and thinking about disease ecology in some novel ways. And so with that, I will turn the time over to Jessica, and thanks very much for joining us.
Thank you, Mark, so much for the introduction. And also, I wanted to say thank you again for the invitation. It has been really awesome, the conversations that I've had a chance to have during this visit. So I really appreciate it, and look forward to sharing a little bit more about my lab and what we've been up to for the past few years. So I'll start broadly by talking about global change biology. We all likely recognize that the world is rapidly changing in many different ways, from things like emerging infectious diseases to changing climate, pollutants in our environment, and also habitats are being lost and modified in unique ways. And so the field of global change biology has emerged as a way to help us to better understand how global change influences these natural ecosystems. And so under this broader umbrella of global change biology, my lab is interested in understanding how environmental change influences host-parasite interactions. And so when we think about host-parasite interactions, one perspective and one direction might be to ask, but what are factors that influence a host's ability to defend itself against parasites? Alternatively, from the parasite perspective, what are factors that influence the ability for these parasites to infect their host? Now, building upon this sort of framework, the field of disease ecology has increasingly recognized the importance of the environment in shaping both the host and parasites and also their interaction. Being trained as an ecotoxicologist, when it comes to the environment, my lab is interested in understanding how pollutants might influence this interaction. And so while this disease triangle framework provides a really nice way to understand how pollutants affect each of these pieces of the interaction, one thing to keep in mind is that these interactions don't occur in a vacuum. For instance, in nature, there are often multiple hosts that interact with each other, and this could influence host-parasite interactions. And then also, there are other factors such as and other stressors like invasive species that may interact with pollutants to influence host-parasite interactions. So all of this to say that a central theme in my lab is to ask, how can we integrate ecological complexity into our understanding of disease outcome? And so this is the central theme that I will talk about first in this talk. So I'll tell you about our efforts at integrating ecological complexity using a tiered approach. To do this, I'll highlight our work with secondary salinization, so sodium chloride. And I'll start first by telling you about how secondary salinization influences simple single-host parasite interactions. Then I'll start working in a little bit more complexity by asking how does salt influence host-parasite interactions, but in multi-host communities. And then from there, I'll transition to asking how might abiotic conditions that vary, how can this affect sodium chloride's toxicity on amphibians and also their susceptibility to disease. So the first part will highlight 3 stories, and then from here, my lab is interested in understanding ecological complexity, but also we're interested in understanding evolutionary implications of environmental pollution. And so there are many different examples of evolutionary responses to pollutants, including things like antibiotic resistance, as well as the evolution of resistance in agricultural settings. And so in my lab, we were interested in trying to find ways to integrate in this evolutionary perspective into this disease triangle framework. More specifically, trying to build on this framework, we asked a few questions, including, can hosts and parasites evolve tolerance to pollutants? And if they can, are there costs associated with evolved tolerance? And how might this then have ecological consequences in influencing this host-parasite interaction? And so in the second part of this talk, I'll highlight our work with integrating in an evolutionary perspective into disease ecology. Specifically, I'll highlight our work. I'll switch gears here from talking about secondary salinization to some of our pesticide work. And I'll talk about 2 parts: plasticity as a mechanism for tolerance to pesticides, and then how this may lead to costs that can influence host-parasite interactions. So this is the outline for today. And with that, I'm going to go ahead and jump right into the ecological complexity part of this talk. So here I'm going to talk about our work with secondary salinization. This is the influx of sodium chloride into freshwater ecosystems. This is an issue worldwide, and there are many ways that salts can enter into freshwater systems, from coastal flooding to the use of road salts. This is something that is really big where we are in New York. And also through agricultural practices such as irrigation, this can bring salts into the soil, and the salt can run off into freshwater ecosystems. And so in this first section, I'll tell you how secondary salinization can influence simple and single host parasite interactions. In my lab, we work with 3 different parasites, ranavirus, trematodes, and chytrid, BD. For the sake of time today, I will focus this talk on just the trematode. So the trematode, the image that you're seeing here, this is one of the life stages of the parasite. It has a complex life cycle, meaning it utilizes multiple hosts. This is the first free swimming stage, the cercaria. And the cercaria infect larval amphibians. And what they do is they encyst in the kidney. So they'll swim up the cloaca of the tadpole, encyst in the kidney. And that image that you see below the tadpole, those are the cysts that these parasites form. This is a dose-dependent parasite. So the more parasites a tadpole has, the higher, the more negative effect the disease. So from this life stage, in order to make it to the next part of its life cycle, it needs to be consumed by a bird or a mammal. And in the gut of the bird or mammal, they reach maturity. These parasites then reproduce sexually and produce eggs. These eggs are deposited into the wetland, and in the wetland, they hatch into the second free living stage, the Myristidia stage. And this Myristidia stage infects snails. In the snails, they produce rediae, and that rediae is what generates the cercaria that then starts the life cycle all over. And so in this first section, the question I'll ask is, does sodium chloride modify host-parasite interactions? To do this, I will focus on just this part of the life cycle between the cercaria and the amphibian. Specifically, I'll talk about wood frogs as a model host and Trematodes Echinostoma tribulus. Throughout this talk, what I'm going to try to do is highlight the graduate student or the student that is responsible for the experiment. This work is done by Nick Buss. He is a 5th year graduate student in my lab and is currently looking for postdoc positions, and he's an amazing student if anybody is interested in a postdoc in the future. So what Nick did to address this question is he exposed wood frog tadpoles to 4 different concentrations of sodium chloride. For reference, seawater is about 19.4 grams per liter. In aquatic ecosystems or in the literature, the highest concentration we found was about 18 grams per liter of chloride, and this was in runoff. In actual wetlands, the highest concentration we found was about 4.6 grams per liter, though the majority of wetlands have ranges of chloride around 0.1 to 1.1 grams per liter. So our concentrations are environmentally relevant, including one concentration that falls into the range that is found in wetlands, albeit on the higher end. And also these concentrations are sublethal. The lethal concentration for wood frogs is 4.1 grams per liter. So using these concentrations, what Nick did was he exposed wood frogs to these concentrations, then added 50 trematodes, and his question was how many of these 50 trematodes successfully encysted in each one of these tadpoles in these different treatments. Looking at the results, on the x-axis here is sodium chloride concentration, on the y-axis is the average number of cysts. Compared to tadpoles that were not exposed to sodium chloride, Nick found that tadpoles exposed to all 3 concentrations of sodium chloride were more susceptible to trematodes compared to that control. And so taking a step back, we found that sodium chloride can modify host-parasite interactions by making tadpoles more susceptible to the parasite. Now, one of the main take-homes, I think, from this study is in the field of toxicology, there's often this emphasis on what are the lethal effects. I think studies like these underscore the importance of also considering these sublethal and lower concentrations of pollutants, which are more commonly found in natural ecosystems, because while they may not cause direct lethal effects, they can still have negative consequences by influencing that interaction. Now, that being said, one of the limitations of this study, as I alluded to in the introduction, is that we only looked at a single host species. In reality, in nature, there are often multiple interacting species that interact with each other, as well as with the parasite. In fact, this relationship between host biodiversity and disease outcome is a major theme in disease ecology. It's a debate, a topic that has been hotly debated for the past few decades, is biodiversity disease risk. And so what I'd like to do in this slide is now move on to the second part of this ecological complexity talk and highlight what the biodiversity disease relationship is and why so many people are interested in this relationship. And so when we think about the biodiversity disease relationship, oftentimes the effect that comes to mind is the dilution effect, or the idea that as species richness increases, individual and overall community infection is reduced. We think this is really attractive because it provides a clear and positive benefit of protecting species diversity. And so if you protect species diversity, there's that benefit of reducing overall infection. The challenge, however, is that while we do see the dilution effect, and there are many studies that show the dilution effect, there are also studies that show no relationship between species diversity and overall infection, and then studies that show the opposite effect, where as you increase in species diversity, you actually get an increase in overall infection. And so my lab got really interested in understanding this biodisease relationship, and in this section, what I'll do is I'll highlight one factor that we think might be contributing to this equivocal nature of the relationship. So to do this, I wanted to first take a slide to talk about the mechanisms driving the dilution effect. To do this, I have generated 2 hypothetical communities. The community to the left is a community with lower host diversity, just a single species, the blue species. The community to the right is a higher host diversity, so 3 species here, the blue, the red, and the yellow tadpole. So looking at the lower host diversity community, in the presence of parasites, if these tadpoles are highly susceptible hosts, then we might predict that the parasites would be successful in infecting their host, so 100% infection here. In contrast, if we look at the higher host diversity community, dilution effect theory would predict that there's a higher likelihood that in these communities, they're going to be hosts that are less susceptible to the parasite, so less competent hosts. In this scenario, parasites might be attracted to the less susceptible host, but for reasons like increased resistance to disease or behavioral traits that allow the host to avoid the parasites, these hosts are not successful in these parasites are not successful in insisting in the host. And so we get a reduction in overall infection in communities with higher host diversity. So graphically, it would look something like this. Now, one of the challenges with these types of studies is many of these studies are conducted in benign environments. This is particularly relevant because, as I just showed you in the previous section, pollutants in the environment can change patterns of disease susceptibility. We showed that sodium chloride could increase tadpole susceptibility to parasites. And so if we're looking at this scenario, if I could draw your attention to the right side of the higher host diversity side, in the presence of sodium chloride, our less susceptible hosts may no longer be less susceptible. They may no longer act as diluters that draw parasites away. Instead, we might see something like this. So even though there's higher host diversity, we may not see that benefit of a reduction in overall infection. And so with that in mind, we came to the hypothesis, and we hoped to, at the point we looked to test the hypothesis that pollutants play an important role in shaping this magnitude of the biodiversity disease relationship. So to address this hypothesis, we had to ask 2 questions. The first, we needed to understand what NaCl, what salt did to modify host susceptibility to parasites. Knowing this information, we can then ask and make predictions about how sodium chloride contamination would modify that relationship. And so again, this is work done by Nicholas Buss in my lab. So Nick went back to the original design, and what he did, instead of just focusing on wood frogs, was he looked at peepers, wood frogs, and toads, and he replicated the design, exposed them to either no salts or salts environments, and then exposed them to 50 trematodes. Again, he looked at the average number of cysts that were found in each one of these species. In this figure, the x-axis are the different species, and what I'll show first are the results in the absence of salt. So when there isn't salt in the environment, peepers, wood frogs, and toads, they're all similarly susceptible to the parasite. When we added salt, we found that peepers became 34% more susceptible to the parasite, so similar to that response we found in the first study. We found that wood frogs became 64% more susceptible. And for toads, it didn't matter whether there was salt in the environment or not. They were similarly susceptible to the parasite. And so now that we understood, and we had data, at least preliminary data, for how sodium chloride could influence host susceptibility to parasites, we can then think about hypotheses for how sodium chloride contamination could change the direction or the magnitude of this biodiversity disease relationship. So starting with in an environment without salt, we saw that peepers, wood frogs, and toads all had similar levels of susceptibility. And so we predicted that there wouldn't be a dilution effect. They all have similar levels of susceptibility, so there's no one species that might act as a diluter, pulling away parasites from others. So as you increase in species diversity, there shouldn't be a benefit of a reduction in overall infection. There's no diluter species. In contrast, when we look at salt environments, we can see that toads are significantly less susceptible to parasites than peepers and wood frogs. Potentially, this species could serve as that diluter. And so in salt environments, we predicted that as species diversity increases, specifically any community with a toad, we might see a benefit of overall reduction. So again, our prediction that pollutants might play an important role in shaping this direction and the magnitude of the biodiversity disease relationship. So to test this question, what we did was we generated 7 different communities. 3 single species communities, so only peepers, only wood frogs, only toads. 3 2-species communities, so the combination of the 3 species. And then 1 3-species community with all 3 of the different species. And so what we did was we reared them in either salt or no salt environments. And one thing I wanted to point out, while we did manipulate species diversity, we kept density constant. We exposed these communities to parasites and then counted how many successfully encysted. Okay, so moving on to the results, this slide represents communities exposed to no salt. I've organized the x-axis from lower host diversity to higher host diversity. The first panel represents all communities compared back to a single community, a single species community of peepers. Second one compared back to a single species community of wood frogs. And the last one compared back to just toads. So reminding everybody of the predictions, we predicted that because peepers, wood frogs, and toads had similar levels of susceptibility, there would be no benefit of increased, there would be no benefit when you increase host diversity. So something that looks like this. So looking at the data, for all 3 species comparisons back to the single species, we found no evidence of the dilution effect. So with that, we can move back. In the absence of sodium chloride, we found no dilution effect. What happens when we change the environment to include salt? We know that in our first lab study, it made peepers and wood frogs more susceptible to parasites. It didn't change the susceptibility of toads, but toads were less susceptible to parasites. And so pollutants modified patterns of host susceptibility. How does this affect this relationship between diversity and overall infection? These are results from environments with 1 gram per liter of sodium chloride. So peepers and wood frogs were highly susceptible to the parasite, so we predicted that they would have high susceptibility. Toads were less susceptible, so we predicted that any community with toads would have a reduction in the number of cysts in that community. In our most diverse community with the 3 species, we predicted that toads would also cause a reduction in overall infection, but because there were only, because we held constant density, there were only 2 toads compared to 4 highly susceptible species. So we thought that the reduction would not be quite as dramatic. So looking at what we found, compared to single species peepers communities, we saw a significant decrease by 28% when toads were added to the community. Compared to single species wood frog communities, we saw a significant decrease when toads were added by 45%. For the most diverse community, compared to the single peeper community, there was a 30% decrease. Compared to the wood frog, there was a decrease, but this was not significant. So taking a step back, I know I just presented a lot of data. And so in this next slide, what I'll do is put it all together. So originally, we were interested in, as a lab, in why this relationship varied so much, and we hypothesized that pollutants were important in driving this, this relationship variation. And so we found that sodium chloride exposure altered host susceptibility to parasite, and this alteration caused a shift, this shift in host susceptibility modified that biodiversity disease relationship. One thing to keep in mind is that this study focused solely on secondary salinization, so sodium chloride. We know that there are tons of different chemicals out there that can have different effects on host susceptibility. But I think the main take home here is regardless of what pollutant is of interest, we think that the equivocal nature of this biodiversity disease relationship can be influenced by pollutants. So with that, thinking ahead, what might this mean in terms of management implications? We know that diversity is a really important ecosystem service and is something that should be protected and should be conserved. But I think what this study suggests is that we also need to consider context. Diversity is not necessarily always a fix-all. So overall, I think this study highlights the importance of incorporating this global change perspective into our biodiversity disease studies. So with that, we're moving on to the third part of this section. Thus far, I've told you a little bit about how sodium chloride can influence single species interactions with their parasites. We've talked in the last section about how adding multiple hosts can modify host-parasite interactions and change the influence of salt. In this third section, what I'd like to talk about is highlighting the recognition that organisms are embedded in ecosystems that may vary in abiotic conditions. And importantly, this variation in abiotic conditions may have variable effects on sodium chloride. So they may exacerbate or even dampen the effects of salt. And so in my lab, thinking in this third section, what we'd like to talk about, or what I'd like to talk about is how do shifts in abiotic conditions modify the effects of sodium chloride. One way that abiotic conditions may differ from wetland to wetland or aquatic system to aquatic system is through the input of leaf litter from terrestrial ecosystems. Leaf litter from different species of trees may vary in their chemistry, and when they fall into aquatic ecosystems and degrade, this can have potentially a shift. It can cause a shift in the abiotic conditions of the aquatic system. Specifically in my lab, we're interested in how shifts from native plants to invasive plants, how this might change the water chemistry as invasive plants take over and drop their leaves into aquatic systems. How do shifts in abiotic conditions caused by invasive plant leaf litter affect susceptibility to sodium chloride? And then secondly, continuing on the theme of host-parasite interactions, how might invasive leaf litter modify host-parasite interactions? This work in this section, this is work done by Dr. Devin DeJacopo. He was a former PhD student in my lab, and it was done in collaboration with Dr. George Meindl. If any of you were in my talk yesterday, Dr. George Meindl and the Hua Lab have a collaboration where we provide course-based undergraduate research experiences. So this research was done by a class. So what we did was we went out to a wetland and collected leaf litter from 6 different species. In white outline, this is the native species. In red outline, these are the invasive species. We chose species pairs based off of where they were found in the wetland. We took the leaf litter and soaked them in water for 28 days. And from there, we filtered out the leaf litter and put the leachates into glass beakers. In each one of these beakers, we placed a single embryo, and we put an embryo of either a leopard frog, which is native to our area, or an African clawed frog, which is non-native to our area and invasive in some parts of the United States. So again, the question here is, how does being raised in different abiotic conditions, here our abiotic conditions are related to native versus invasive leaf litter, how does being raised in these different conditions influence a tadpole's susceptibility to sodium chloride? So we raised these embryos in these solutions until they reached the tadpole stage, and then we conducted a toxicity assay. In this figure, on the x-axis are the plant species, native or invasive. On the y-axis is average time to death. In yellow will be leopard frogs. In red will be the non-native African clawed frogs. You may have noticed we did test 6 species, but for the sake of time and also because our principal components analyses indicated that our native leaf litter, like the chemistry was more similar within native leaf litter than were different than invasive leaf litter, I made the decision just to separate them out for this talk. So what did we find? We found that for leopard frogs, being reared in invasive plant litter made them less tolerant to sodium chloride. So something about the invasive plant leachates made these leopard frogs more susceptible to sodium chloride. In contrast, we were really surprised to find the opposite effect for African clawed frogs. For African clawed frogs, being raised in invasive leaf litter actually made them more tolerant to sodium chloride. And so we were really curious about this effect because it appeared that these invasive leachates facilitated the non-native African clawed frog, but had negative effects on the native leopard frog. And so we wanted to dig into this a little bit deeper. And so specifically, we wanted to ask, why does invasive leaf litter make leopard frogs more susceptible, but African clawed frogs less susceptible? To do this, we looked a little bit deeper into the water quality and the leachates. We conducted a wide range of analyses. I won't talk about all of these today. Instead, I'll focus on general hardness. I'm focusing on general hardness, which is calcium and magnesium, because we found a significantly higher concentration of higher general hardness in invasive leaf litter compared to native leaf litter. And this stood out to us because we know that the general hardness preference of leopard frogs is generally lower than for the African clawed frogs. And so we hypothesized that perhaps African clawed frogs did better in invasive leachates because it more closely resembled their preference, whereas for leopard frogs, it was a higher general hardness that perhaps was stressful, making them more susceptible to pollutants in the environment. So we conducted a second study, and in this one, we just manipulated calcium chloride concentrations. We had low and high concentrations to mimic native concentrations of leachates and invasive leachates. So that figure to your left, this is data that you already saw. In the figure to the right, this is the new experiment that the students in the course conducted. And what we found, the results closely resembled the original study. And so this suggested to us that general hardness is likely one mechanism potentially contributing to why leopard frogs are more susceptible to salt when raised in invasive leaf litter, but the opposite for the African clawed frogs. All in all, I think that the main take home here is that as human populations continue influencing natural ecosystems, considering how abiotic conditions that arise from these environmental change interact, I think will become increasingly important. So we show that the toxicity of sodium chloride is influenced by shifts in abiotic conditions. So what about host parasite interactions? Does invasive leaf litter modify host parasite interactions? What Devin did was he went out and used 2 pairs of native and invasive leaf litter. And this time working with American toads, he raised these toads in the leachates, and then he moved them to clean water where he exposed the tadpoles to the parasite. And so he decided to move these tadpoles to clean water because it was his goal to isolate the effects of the leachates on the host. And we actually do have, we did the reverse where we isolated the effects of leachates on the parasite, but I didn't have time to include that today. But if anybody is interested, I'd be more than happy to talk about that side of the experiment. In this side of the experiment, what Devin found was that for both pairs, tadpoles that were raised in invasive leaf litter leachates were more susceptible to parasites compared to those that were raised in native leaf litter. Again, underscoring the importance of considering potentially the interaction between multiple global change factors. So thus far, I've talked a lot about how my lab has worked to integrate in more and more ecological complexity. Where do we hope to go from here? We're actually hoping to zoom in a little bit more to understand what are the mechanisms. Why are some tadpoles more tolerant to contaminants than other populations of tadpoles? And so one of my graduate students, Grayson Scheidmantel, has opened up new doors in our lab thinking about physiology. She's interested in measuring how tadpoles that differ in tolerance, how they may be, how their hormones might be looking. So corticosterone, she's interested in measuring oxidative stress metrics. Her work is in collaboration with Ivan Gomez Mestre in Spain. And ultimately, her goal is to understand and improve upon non-invasive metrics, corticosterone indicators, so that we might better pinpoint which populations are more at risk. In addition to this sort of future direction, I think one challenge in ecotoxicology is that here I just spent 35 minutes talking to you about a single contaminant, secondary salinization, sodium chloride. But we know that there are a ton of other pollutants in the environment. And working to increase this ecological complexity for a single contaminant, it took us 5 years to get to this point. So how might we start to make generalizations about the effects of pollutants on host-parasite interactions? To do this, we've been really fortunate to rely on amazing undergraduates in the lab. And so these undergraduates come in with a pollutant of interest, and they sort of design a study asking, what does this pollutant do to host susceptibility to parasites? And they take it all the way from experimental design, looking for funding, all the way to publication. And while it benefits them, it also provides us with quite a bit of data on what these different pollutants do to a host's susceptibility to disease. And so as you can see, those arrows represent what these various contaminants do. It's all over the place. And so where we're hoping to go next is to utilize modeling approaches. Can we predict when these different stressors are likely to lead to increased susceptibility to parasites? So that's what we hope to do as we gain more data on what these various pollutants do. So with that, this is the end of the ecological complexity portion of the talk. We talked about the effects of salt on these different levels. In the next 15 minutes or so, I hope to shift gears to talk more about the evolutionary perspective, the evolutionary arm of our lab. Here, we'll transition to talking about salt to pesticides, and I'll talk about the evolution of tolerance to pesticides. So when it comes to the evolution of tolerance, the traditional approach might be to think about genetic changes and constitutive traits due to natural selection. So you have a population, it's exposed to contaminants. You may have some individuals that have a constitutive trait that allows them to persist. If this trait is heritable, then over time you can get the evolution of tolerance. So while the evolution of tolerance can allow these populations to persist, there are a few limitations. First, it depends on that population having those individuals that have the ability to tolerate the contaminant, that constitutive ability to tolerate the contaminant to begin with. And also, it can take multiple generations. And this multiple generation is an important point, because when it comes to human activities, oftentimes the things like pollutants cause really rapid change. And so with that in mind, I became really interested in exploring whether there were other mechanisms for how organisms can rapidly or more rapidly deal with pollutants. And so this led to us thinking a lot about phenotypic plasticity, or the capacity of a single genotype to exhibit variable phenotypes in different environments. And so an example, one of the examples that I like to share in terms of phenotypic plasticity is the response of tadpoles to non-lethal predator cues. So in the presence of the smell of a predator, these are tadpoles of the same species, of the same egg clutch, and the tadpole on the top was raised in an environment without predators. The tadpole on the bottom was raised in an environment with predator. And the point here is that tadpole on the bottom, within a single lifetime, can induce a trait that allows it to better deal with predators in its environment. And so with that in mind, we became curious, what about pesticides? If they can induce rapidly a trait to deal with predators, can they also induce a trait tolerance that allows them to better deal with pesticides? So can they do this rapidly within their lives? To address this question, what I'd like to do is spend this slide talking about the reaction norm figure, orienting everybody to this figure, because the rest of this section will use this same structure. On the x-axis is environment, either pesticide or no pesticide. On the y-axis is the trait of interest. This is pesticide tolerance. So if a tadpole were to be expected to have plasticity, we might see a reaction norm line that looks something like this, a sloped reaction norm line. In environments with no pesticides, they would exhibit low pesticide tolerance. It wouldn't make sense to have tolerance when the pesticide is not there. However, that same tadpole, if it is plastic, if exposed to pesticides, they would have the ability to increase that tolerance and induce higher tolerance. And so that slope reaction norm line would indicate plasticity. Theory predicts that plasticity is favored in environments where you would have variable pesticides. So organisms that would not exhibit resources or not expend resources to tolerance if it's not there, but have the ability to induce tolerance if the pesticide were to show up. So variable pesticide environments, we should expect plasticity. In contrast, in constant pesticide environments, theory predicts that over time, due to the cost of plasticity, we would see this loss of plasticity. If the pesticide is always there, it makes sense to always just invest in high pesticide tolerance, constitutive tolerance. And so this process is called genetic assimilation, where an environmentally induced phenotype becomes fixed. And so for me, this was really exciting, and why we're so excited about genetic assimilation is because it provides another mechanism for evolved constitutive tolerance. The traditional paradigm can lead to the evolution of constitutive tolerance, but so can genetic assimilation. While we know a lot about the traditional paradigm and the evolution of tolerance, we know much less about genetic assimilation as a contributor to the evolution of pesticide tolerance. And so that's the second question that we'll ask in this section. To put it all together, we're going to ask, can tadpoles induce tolerance to pesticides? We're looking for a sloped reaction norm line here. Does genetic assimilation contribute to the evolution of pesticide tolerance? One way to do this would be to take a plastic population, change the environment to a constant pesticide environment, and see if over time it reaches constitutive tolerance. This can take a lot of time, and so instead what we did was we substituted space for time. So we went out on a gradient, a distance from agricultural gradient, far to close from agriculture, and we took populations that were far from agriculture, which don't experience pesticides as constantly, and these would serve as our ancestral populations. Populations close to agriculture would serve as our derived populations. Those that are more commonly exposed to pesticides, more constantly exposed to pesticides. So using these different populations across time, we could simulate time. So populations far from agriculture, the ancestral populations should have plasticity. Populations close to agriculture, or the derived populations, should have constitutive tolerance. If both these criteria are met, we would say it's consistent with genetic assimilation. So to test this, we went out to 15 populations in Pennsylvania, and we worked with 15 populations of wood frogs. They vary in their distance to agriculture, as well as you'll see their tolerance. So we measured their tolerance across these 15 populations. So what did we find? On the x-axis, this is the environment again. On the y-axis is tolerance. The first question, can tadpoles induce tolerance to pesticides? Here we're looking for that sloped reaction norm line. In that first panel, we see the gray lines represent each population. The black line represents the average, and we do see, and we did find several populations where tadpoles exposed to 48 hours of pesticide were able to induce increased tolerance to the chemical later in their lives. So this is evidence that plasticity may be a mechanism allowing these tadpoles to persist. Does genetic assimilation contribute to the evolution of pesticide tolerance? Again, we're looking for that flattening out of the line as you move closer and closer to agriculture or derived populations. This first panel represents 5 populations that are far from agriculture. And as we move closer to agriculture, you see that, that line flattening out, suggesting a shift from inducible tolerance to constitutive tolerance as you move closer to agriculture, which is consistent with the evolution with genetic assimilation theory. So all in all, I told you this data, I showed this data as a foundation because the work that we do with host-parasite interactions, it builds off of this finding that pesticides in the environment can lead to hosts exhibiting 2 types of tolerance, inducible and constitutive tolerance. We know that these different mechanisms for tolerance are likely associated with different costs. And so we know if pesticides are not the only stressor that organisms face, how might exhibiting these different mechanisms of tolerance lead to their ability to avoid parasites? And so in this final section, what I'll talk about is the link between evolutionary processes and ecological interactions. So the question in this final section is, does inducible versus constitutive tolerance to pesticides influence host parasite interactions? This is work done by my former PhD student, Dr. Vanessa Werthner. She is now at the EPA. She's doing incredible work with pesticides now there. What she did was she went out to the same 15 populations of wood frog, and she brought embryos back to the lab and reared them in common garden environments and tested their susceptibility to the trematode. What Vanessa found on the x-axis is a gradient from constitutive to the left to inducible on the right, and she found that tadpoles that exhibited inducible tolerance were more susceptible to the parasite compared to those that exhibited constitutive tolerance, suggesting that there are differential costs associated with these different pesticide tolerance mechanisms. Vanessa is interested in understanding questions about the microbiota and also interested in understanding a little bit more about what might be driving this pattern here. And so this caused her to think a little bit more about why inducible populations were more susceptible to the parasite. And these parasites find their host via by chemosensory techniques. And so what she thought was that variation in skin microbiota might lead to differences in how successful hosts or how successful parasites are in finding their hosts. And so in collaboration with my good friend and Dr. Obed Hernandez Gomez at Dominican College, what Vanessa did was she went out to the populations and again brought them back to the lab and reared them in lab environments. She swabbed their skin, and what she found was that on the x-axis here, you have distance to agriculture, but those that were close to agriculture, again, has constitutive tolerance. Those far from agriculture had inducible tolerance, and again, those were the more susceptible populations. She found that those that were inducible also had a higher alpha diversity of the skin microbiota. For us, this is just preliminary. We're interested in pursuing further whether this truly is a mechanism for the parasite looking for their host. Excited to follow up on this. Really sad that Vanessa has graduated because now we'll have to look for somebody else to follow up on this work. So with that, we show that these different mechanisms can have cascading consequences on susceptibility to parasites. But to round out this disease triangle, triangle, what I'll talk about in this last section is, what about the parasite? We know hosts can evolve tolerance and can exhibit constitutive and inducible tolerance. What about the parasite? And so this is work that is done in collaboration with Dr. Jason Hooverman and Dr. Rick Relier. And what I'll tell you about is actually this part of the life cycle. So we didn't use great blue herons, we actually used an avian host, a chick host. What we did was we went back out to populations that of known parasites that vary in their distance to agriculture. We collected 6 populations, and we wanted to know how these different populations varied in their tolerance to pesticides similar to their host. We measured hatching success, survival, and the ability to induce tolerance. In this figure on the x-axis, I've arranged the populations from close to agriculture to far from agriculture. We wanted to know if there was evidence that parasites might evolve tolerance to pesticides, and we're measuring hatching success here. We raise these parasites as eggs in the chemical. We use chlorpyrifos here, and we hypothesize that if there is evidence for evolved tolerance, those from populations that are closer to agriculture would have higher hatching success. They are more tolerant to the chemical. I'll note that this is preliminary data. This is something that we were able to collect right before COVID hit. So we did find a relationship here. It seems that parasites near agriculture do appear to be more tolerant to the pesticide. I don't have time to show you the inducible tolerance data yet, but thus far, we're finding that at least some populations of parasites can induce tolerance to pesticides. So when reared in pesticide environments early in their life, they can become more tolerant later in their lives. So with that, it seems that trematodes and parasites in this triangle may also have the ability to exhibit tolerance via different mechanism. The next step that we're hoping to ask then is how then does this then influence the parasite's ability to infect the host? So with that, I will put everything together and in what we do in our lab. In my lab, we're interested in the effects of pollutants on the environment. We're interested in how it affects host-parasite interactions. From an ecological perspective, we're interested in how pollutants influence various organizational levels, from host to multi-host to ecosystem levels. And from the evolutionary perspective, we're interested in how hosts and parasites evolve tolerance and how this might influence their interaction. And lastly, as I noted yesterday, a major component of our research is science communication and outreach. And we do this at the university, local, and scientific community. With that, there are many people and organizations to thank. These are the funding sources here. These are just images of some of the members of my lab. They are the ones that did this work. I always say I'm just so lucky that I get the opportunity to share the research that they do. So with that, I'd be happy to take any questions. Thank you so much for spending your evening with me and listening about our lab's research.
Great. Thank you very much, Jessica. And I want to tell folks who do have questions, we're going to use the chat feature. So, so just if you have a question, type it into the chat, and then I will ask it for and present it to Jessica. And while you're doing that, while you're thinking of questions and getting your typing fingers warmed up, I'm going to ask a question that's a little, basically what I wanted to say was that you have the most amazing illustrations in your presentations of anybody I've seen. And I'm wondering, is that all because you've been lucky enough to find the students who do that, or do you have access to illustrators? That how can people in our audience be able to do things like this?
So if you're asking about the images, all of the images in this presentation are from the artist Ben McLaughlin that I talked about his work yesterday. Ben is actually in graduate school right now at Rensselaer Polytechnic Institute, not Rensselaer, Rochester. And he also has services that he would be more than willing to make these images of your research. And so please hire him. He is fantastic to work with. And hopefully I've shown that the images do help with science communication. At least it helps me.
Mm-hmm. Great. Yeah, our first chat was not actually a question, but a comment from Brian saying that Ben rocks. So